Sydney, you have some presenters coming, is that correct? Or are you presenting for the whole group? Hmm. All right, well, shall we go ahead and get started? Hi, Courtney.
All right, so I guess I can go ahead and screen share.
Hold on just a second.
Okay.
If you don't mind, I'm going to go over a few things. So first of all, welcome to the third session of the Showcase of Undergraduate Scholars. And this is the medicine session. Just a few housekeeping things to go over. Just as a reminder, this session is being recorded. We do encourage our audience and our students to ask our presenters questions. You can use the Q&A feature that's located on your toolbox. I will tell you some of the Q&A, it works on the students features. Some you have to use the chat. So check both areas after you're presenting. And I think that will do it. Do students have any questions? Oh, minimum of 10 minutes. That does include your presentation and your Q&A. Okay, so try to keep it within that if at all possible. Okay. All right. So our first presenter is Courtney Martin, and Courtney works with Dr. Nathan Vanderford in the College of Medicine, Toxicology, and Cancer Biology. All right, Courtney, are you ready?
Yes.
All right.
Let me try to do this here. Okay, you all might have to tell me if you can see it. Can everyone see it? Okay, perfect. Yes. So hello, everyone. My name is Courtney Martin. I'm a senior biology major here at UK. And the title of our project is Investigating Cancer in Appalachia through Content Analysis of Oral History Interviews. I just want to start by giving a shout out to my mentor, Dr. Nathan Vanderford. It has been such a pleasure and honor to be able to work under him and learn under his mentorship. So I appreciate him and, of course, the Office of Undergraduate Research, Evie and Jesse, for all they do. So thank you all. And we'll go ahead and get started. So cancer rates are high in the US, but among the entire nation, it's actually Kentucky that ranks first in cancer incidence and mortality. Kentucky experiences 26,000 new cancer cases each year. However, it's the Appalachian region that experiences the greatest disparities among Kentucky. For example, lung cancer death rates are almost double in the Appalachian region compared to the national average. In addition, Appalachians are 8% more likely to die from a preventable or a screenable malignancy. These disparities necessitate further investigation and action. The purpose of our research is to better understand the reasons giving rise to high rates of cancer in the region and the potential solutions to addressing those problems. To do so, we recruited individuals from within the community with cancer-related experience. We took a community engagement approach and we recruited oncologists, those working in cancer-related fields, as well as those with cancer-related experience, whether it was their own diagnosis and or the diagnosis of a family member. Each of these participants sat for an individual oral history interview, which averaged about an hour and 20 minutes in length. The topics of interest during these interviews include problems giving rise to high rates of cancer in Appalachia, potential solutions to addressing those problems, as well as the future in terms of success and what the future may look like without intervention. From the data that we collected, we analyzed it using qualitative content analysis, and the data was condensed into themes, subthemes, and subtopics, respectively. Some of the themes that emerged during our analysis can be seen here in the results section, and these include lack of access to healthcare, distrust of outsiders, including healthcare providers, as well as lack of education and educational barriers. So Appalachia is known for high levels of poverty. As such, the lack of infrastructure means limited local healthcare facilities. And unfortunately, this makes care inaccessible to many. Patients who are able to commute find themselves traveling 2 to 3 hours to the nearest healthcare facility simply to get the care that they need. Nonetheless, this results in inconsistent appointments, which means delayed treatment and ultimately poor cancer outcomes for someone with a cancer diagnosis. Beyond that, many physicians that currently practice in Appalachia often aren't from the region. So if you see the distrust here, that comes about when there's this barrier between engaging with the healthcare system. Appalachians simply do not trust outsiders. They worry about being talked down to or feeling less than. They're conscious of their accents and dialects. And again, this really causes a barrier when it comes to engaging with the healthcare system. So what can be done with all of this information? Well, immediate solutions would include utilizing telemedicine as well as technology to bridge the gaps in accessibility. Obviously, long-term, we would want to ultimately increase the number of local healthcare facilities in the region, but more immediately, we would use things like telemedicine and expanding transportation services to help patients get to and from their appointments. Further, there's a lot that needs to be done in terms of education. So there's a lot that can be done with education and decimating it. We could decimate it to the community through webinars and seminars, which would also, need government intervention to help us create policies and programs for getting that information to the community. And another way that other literature has shown to be successful in terms of getting cancer education into the community is through integrating that into the curricula of schools. So there was a study done on middle school and high school students, and it showed that an educational intervention did increase cancer literacy. So that is one way that we saw through our themes as well in terms of addressing the problem at hand. And lastly, I would like to talk about the lack of physicians and distrust. So in terms of the lack of physicians, this problem arises because of the lack of poverty, because of the lack of opportunity. So we really want to recruit and retain physicians. And one way to do that would be through incentive programs to encourage them to come to the area to practice medicine. And lastly, distrust. So like I mentioned previously, distrust is a barrier when it comes to engaging with the healthcare system. And one way that our participants propose to address this is through integrating through integrating Appalachian culture into the onboarding preparation of healthcare providers. So really just giving them that foundation and that background of Appalachian culture so they can best assist patients, and in our case, patients that may be dealing with cancer. So in conclusion, there's a critical need for addressing these issues. As mentioned there at the beginning, cancer rates are substantially high in Kentucky and even more so in the Appalachian region of the state. So definitely an area that we need to focus on addressing. And through our project, we have been able to identify many areas and means of intervention through community voices and through people who are living through, the cancer epidemic in the area. But nonetheless, a lot of these solutions would require legislative action. As mentioned, we need a lot of government intervention to create and develop policies and programs to implement to help us really address the problem at hand. Nonetheless, though, these solutions that we have found through our research and through related literature does have the potential to help address these problems and ultimately improve the cancer outlook in this area. As an Appalachian native myself, the cancer epidemic affects my family, my friends, and my community. And I hope through the information that we've gathered, we're able to long-term implement these solutions and follow them, over a period of time to determine their impact and hopefully help to continue to improve the cancer outlook in the area. And these are just some acknowledgments regarding funding. Thank you all, and I'm happy to answer any questions that you have.
Nice job, Courtney.
Thank you.
Very relevant subject. Hey, as we're waiting for questions, could you tell us, how did you get started in this research?
Yeah, absolutely. So I was actually in Dr. Carpenter's class. She's in the, it was a writing and rhetoric class, and she introduced me to Dr. Vanderford. I was really interested in like advocacy and prevention, and really just wanted to learn more about my community. And she introduced me to Dr. Nathan Vanderford, and I applied for one of the fellowships through the Office of Undergraduate Research, and really we just started working from there. And since then, I've just really found my passion for research in the field and the area that I like most, and really just learning more about Appalachia and how I can give back to my community has been wonderful. So really happy to be able to work with Dr. Vanderford.
Awesome. Does anyone have any questions for Courtney? All right, Courtney. Well, just to conclude this, what are your plans after this semester?
Yeah, absolutely. So I'm not graduating until December, and then I'll be back in the spring, just getting some more prerequisites that I need for med school that don't count towards my degree. But yeah, after the semester here, I want to continue doing research. I'm hoping to work with Dr. Vanderford over the summer on a related topic about cancer in Appalachia. And yeah, I just want to continue really doing research relevant to Appalachia, and then long term, I hope to go to medical school in the future.
Awesome. I actually think that you do have a question in the chat,
Okay.
actually in the Q&A. Can you see that? And if not, I will read it for you.
What are some of the easier things that can be addressed? That's a good question, Luke. So in terms and Luke asked what some of the easier things are that can be addressed. So I think obviously lack of access is a big one, and because Appalachia does experience such high levels of poverty, it's not necessarily easy to say let's increase the number of facilities, let's bring all these doctors here when there aren't a lot of opportunities. So those are things that will require a lot of effort and be, sort of long term. But some of the easier things I think would be the webinars and seminars, because a lot of times, and even some of our oncologists that we had said this, people aren't really, they lack health literacy or cancer literacy, which is essentially health-based knowledge. So they aren't really aware of prevention. They don't necessarily know that when I get this age, I need to get a colonoscopy, or if someone in my family had breast cancer, I may need to get a mammogram sooner. So I really think just disseminating information to the public through webinars, seminars, through implementing it or integrating it into the curricula of schools, really just education from a broader side, I think would be some of the more immediate and some of the easier solutions, so that we can get people getting out. Because the longer you wait, the later the stage in which the cancer is diagnosed, we all know means, you know what I mean, it's harder to treat. So I think really education would probably be some of the easier ways to address the cancer epidemic. That's a good question. Thank you, Luke. All right. I think that is all, Evie. So I will stop screen sharing, but thank you all so much. And thank you, Evie, and the Office of Undergraduate Research. I'm excited to hear about y'all's work.
Thank you. Great presentation. All right. Let me see. Can you guys see this or no?
No.
Okay. Technical difficulties again. I apologize. Well, all right, can you see that?
Yeah.
Yes.
Okay, perfect. So this next presentation is going to be by a large group, and we actually are going to give them 20 minutes because they are going to kind of tag team on this presentation. So Sydney, are you going to share screen?
Yes.
Okay.
Okay. Hi, I'm Sydney, and our lab team is excited to share how we have been using Datasonification as a virtual authentic learning tool in molecular biology. This is a new project we've been working on with an interdisciplinary team from the College of Medicine and School of Music. We live in an age of unprecedented biomedical knowledge and advances, as well as a growing interest in science by the general public. This is not surprising, as biotechnology covers nearly every aspect of our lives, whether it is genetically engineered foods, biofuels, new drugs, or materials. Due to advances in biomedicine, it is possible for everyone, for a surprisingly small fee, to have their individual DNA sequenced using products such as 23andMe. In addition, with the ongoing COVID-19 pandemic, we are constantly hearing stories of different variants, vaccines, and testing procedures. While the thirst for scientific news is at an all-time high, it is also a time for widespread misinformation, thus necessitating greater scientific literacy in general, but especially in the biomedical field. Despite this rapid growth in STEM fields, people of color, people with disabilities, women, first generation, and low-income students are underrepresented in the workforce. So that leads to the question, what are some best practices to engage students and to minimize these gaps in STEM literacy and recruitment? To increase inclusion and interest in STEM, authentic learning experiences are hands-on learning opportunities that expose students to real-world applications and supplement classroom education. Authentic learning experiences are proven to facilitate social integration and help students to develop additional skills, including professionalism, teamwork, and independence that will prepare students for future undergraduate study and careers in STEM. However, opportunities for authentic learning have been limited by remote instruction. So how can we engage students in understanding these biomedical concepts using authentic learning experiences while we all work virtually, but also at an introductory level? So before we address this question, Taylor is going to review some of these introductory concepts with how they are often introduced in classrooms today.
Hi everyone, I'm Taylor. So I'm just going to get into a little bit of the biology background. So the central dogma of biology is DNA to RNA to proteins. Proteins are formed by amino acids, and they are structurally and functionally the most diverse and dynamic molecules. They play key roles in almost every biological process. So the order or sequence of amino acids in a protein is what determines the structure of the protein. So interactions between individual amino acids in the sequence cause the protein to twist and fold into a three-dimensional shape. The shape of the protein then determines its function and what the protein is able to bind to. So the unique binding affinity of each protein and how proteins interact with each other is how cell signaling takes place to coordinate cellular processes through a complicated network of interactions. So the leading question for our research project is, how can we make these abstract molecular biology concepts resonate with students? and we chose to use Datasonification. So using a multidisciplinary approach, we integrated arts into STEM, which can help connect students to STEM fields and increase student confidence and creativity. So Emme is going to take over now and help describe the methods of Datasonification.
Thanks, Taylor. Datasonification is the method of converting information to sound. For our project, we are converting complex patterns and disruptions of patterns due to diseases of amino acid residues of proteins into sound, to better demonstrate and understand the molecular basis of diseases, and as a result, change the protein structure and function. Next slide, please. All right, thank you. Datasonification method starts with a student researching a particular disease and identifying a particular protein that plays a role in that. A student would then go to an online databank to access the research information on the protein and download the primary sequence of the protein. These are actually the same databanks that researchers often use. The primary sequence is then uploaded into the synthesizer, and the individual amino acids are assigned notes and beats based on their chemical affinity towards water. For example, those with hydrophobic or nonpolar amino acids that dislike water actually are assigned lower notes and a slower beat. Amino acids that are hydrophilic or polar amino acids that like water are assigned a more higher beat and a more higher note. Now that the sequences are ready for playing, I'm going to turn it over to Andrea.
Hi, I'm Andrea, and we're going to be looking at ALS, or amyotrophic lateral sclerosis. This disease is a motor neuron disease that causes loss of muscle trophism. It is also an example of a disease that is caused by mutated proteins. Mutations in the enzyme superoxide dismutase, or SOD1, have been linked to ALS. They cause protein aggregation and misfolding. Mutant G85R, where glycine is replaced by arginine, causes loss of hydrogen bonds, which affects zinc geometry and ultimately leads to destabilization and aggregation. Here's an example of the sonification of a mutated protein. To better describe how a mutated protein changes cellular communication and cell health, our high school researcher, Lordina, will introduce our broken symphony analogy.
Going back to the diagram shown earlier, this is a simplified model of the calcium signaling. Many different proteins must interact properly with each other for the cell to function normally. This is like a symphony. Basically, this would be like all symphony sections playing in tune with each other. But sometimes in disease, mutant proteins do not effectively communicate with other proteins. The cellular symphony of the protein-to-protein interaction is disturbed. In the context of the orchestra, this would be like one instrument section playing in a dissonant tone compared to the rest of the sections, describing the quality of the overall sound, symbolizing how an individual mutation can impact the cellular health. An epileptic Appalachia, also known as EE, is a disorder that begins in infancy, which is associated with generalized seizure activity and progressive neural impairments. While many mutations likely play a role, a severe lysine to threonine mutation, shown in the red crystal structure, occurs at residue 552 in the potassium voltage-gated ion channel, shown in gray, which is disrupted by the binding interaction of the calmodulin, a calcium-sensing protein, shown in the green, that regulates the activity of the ion channels. The protein-to-protein interaction disruption as a result of the mutation causing the neural hyperactivity seen in EE. In this clip of the Datasonification sonifizer, instrument A is a healthy potassium voltage-gated channel. Instrument B is an EE mutation, and instrument C is an interaction calmodulin protein. After the mutation occurs, the key tempo becomes offset, representing how the mutation disturbs the channel hex. Can you play the sound, please? By placing the mutant potassium ion channel in the cellular context, the musical dissonant is even worse than in the previous example shown how the mutation in one protein can affect the overall cell. To share with you what else we've been working on and to wrap up our presentation, here's Kayla.
So on this slide, we have more examples of diseases we've been studying. While some are neurodegenerative, others pertain to connective tissue. As you can all see, we have been studying sickle cell anemia, Charcot-Marie tooth disease, epidermolysis bullosa simplex, osteogenesis imperfecta, Parkinson's disease, which will be discussed more deeply in another talk we'll be having tomorrow, and of course, other channelopathies. And we wanted to show you more examples to show how there are a variety of diseases that can be studied using our research methods. Because with so many diseases out there, our data sonification can be used for many of them. So ultimately, it is up to the student to decide what disease they would like to study at the molecular level. As for the future, our synthesizer will have different note structures, scales, and tempos to depict biological complexities. We plan to eventually make the synthesizer more user-friendly to enable widespread use. We would also like to spread its use to partnering middle and high schools that we have through our Outreach Start program. By allowing many students to use the synthesizer, we would be able to evaluate students' attitudes towards the STEM field, and hopefully we could also observe more STEM engagement with students. Next slide. Overall, our data sonification simulator provides a STEAM, science, technology, engineering, arts, and mathematics learning experience for students involved in molecular and cellular biology. Not only will students be heavily engaged in their field of interest, but they will also be using actual research data banks, as well as developing creative processes by combining music and scientific concepts. With this aspect of the project in mind, this truly gives students a way to be creative with their studies. And as we continue to pursue this project as a team, future development will enable widespread use at varying levels of complexity, thus providing students with an enhanced learning experience. Next slide. We would like to close out by acknowledging our interdisciplinary team, as well as thanking our faculty mentor and faculty collaborators from the School of Music. And finally, we would like to recognize and thank our sources of funding for our research project. So if anyone has any questions, the floor is now open for them.
Great job, guys. Very interesting. I have a question. So was the School of Music, were they hesitant at all to collaborate with you guys? Was there some convincing you had to do?
Sure.
Actually, oh, sorry, Sydney, were you gonna go? Okay. Actually, they were really excited too, because I actually went to STEAM along with Lordina, and having a STEAM education, which is science, technology, arts, engineering, and mathematics, is like super important. And like, I think that like they were like really like grateful to like, you know, bridge the gap between the sciences and the arts.
This is a great example how bridging different disciplines together can really be creative research. So great job, guys. Let's see, you do have some questions. Can you read them? They are in the Q&A. And if you cannot, I will read them for you. Just let me know.
Can you go ahead and read them?
Okay, sure. Absolutely. Aubrey says, great job. How soon do you think this data sonification simulator will be used in high school curriculum?
So I'll answer that. Thank you, Aubrey, for your question. This is still in development, as we mentioned, with our future direction slide. We want to make it so it is easier to disseminate for widespread use and making it into more of a concise, compact learning module for students to use. And so one thing that we will be looking to do in the future is to involve more teachers in doing that part. So not necessarily extremely soon, but eventually we would like to use this project as a learning module for students. But Lordina is a high school student currently, and so she has joined our lab and has kind of what we have done to put this together is essentially what other students would do. So I guess you could say we are already doing it now, but it hasn't been distributed widely.
So there's 2 more questions. The next one is from Dr. Hannan, and he asked, besides music, is there any other art platform that can be used?
I can answer that one. So as of right now, we haven't really looked into using any other art platform. I guess because we use like the notes and assigned a different note and sound for each of the hydropathy values for the amino acids, we could also maybe do something along with colors, maybe for students that have hearing impairments or something like that, so that they could see the proteins in a different way or the amino acid sequences. But as of right now, we haven't really looked into expanding it past the music right now.
And the next question is from Kara Tower. She asked, what inspired each of you to pursue this project? Andrea, do you want to take the lead on that?
Yeah, so we know that it's been really hard with COVID right now. So I think this was the perfect project for us to all get involved in. And for me personally, I think it's very valuable when you can present information to students in a more tangible, less complex manner to, get them interested in science and also make it a little bit fun, because, who likes to sit in a boring class? Everybody likes, to have fun. So I think this was perfect for that.
Oh, I want to add on to that. And as a student, I feel when I was brought onto this project and I heard how everything was going to go, this project is really interesting, and it would help a lot of high school students, middle school students to really get engaged with diseases and stuff. So we talk about the certain diseases in high school, like Huntington's disease. If a teacher is able to pull this project up and show these students, hey, this is what the disease sounds like. Do you hear that mutation in the sound? That's really going to be helpful for them to really be like, oh, that's the sound. Let me type up another disease. Let me hear a little bit more, and it's going to help these kids to get engaged and want to get involved in the STEM field, while also connecting music to it.
Sydney, do you have anything to contribute?
I was just going to add that I was the Office of Undergraduate Research. I had a summer fellowship with them last summer, and so kind of once everything got moved online because of COVID, this was a great way to transition and still have a really meaningful experience and learn about molecular biology.
Emme?
The only thing that I really want to contribute is I'm a musician, and I also really like science, and oftentimes I feel like, I can't really combine my 2 interests, and so this project was just perfect for that. So
Taylor?
So when I was brought onto this project, we hadn't quite gone into the educational side of it first, or yet, I guess. So the end kind of game for this whole thing would be to find ways to synthesize new proteins with similar binding affinities. And I thought that was really interesting. So it's cool that we've found a way to kind of integrate this into education and make it into something that like students can use to like get more interested in proteins and diseases and structures and all that kind of stuff.
Nice. Kayla?
Yes, so I was actually appointed here by my honors advisor because I told her I was interested in pursuing undergraduate research, and she already knew my interests with genetics and how it would pertain to this project. So she sent me this way, and I had a meeting with Dr. Bradley, our mentor, and I absolutely loved the project that he described because, like Emme, I'm also a musician, or at least I used to be, and I really love music. And then there's also proteins, and I really love learning about proteins because that's all genetics is about. And I really feel like this was a super exciting opportunity to really dive deeper into interests as well as work with a team on a project that's so unique and so new. And ever since I found out about this project and I started getting involved, I've been telling all of my pre-medical friends about, hey, this is what we're doing. And they all agree that this is a really great way to learn about proteins and sequences and genetics and just everything with molecular and cellular biology. And since this is a really difficult subject, I feel like using this method, it makes it a little easier for students. And I would love to also contribute to allowing that to be released to other places so everyone can experience it.
Nicely done, ladies. Did I miss anyone? Did I? Okay, perfect. Any other questions before we move on? Fantastic presentation. Should be very proud. All right. Wait 1 second. There might be. Nope. That's it. So thank you, ladies, very much.
Thank you, Abby.
All right. Our next presenter is, can you see the screen? Lauren. And Lauren works under the direction of Dr. Nathan Vanderford as well. Lauren, are you ready to present?
Yes, I am.
Okay. Let me. Okay.
So my name is Lauren. Some of you may know me by Kayla, but the study is using culturally focused storytelling to empower Appalachian Kentucky youth to understand and address cancer disparities in their communities. We have no conflicts to disclose. But basically by the end of this study, or by the end of this presentation, I would like for the audience to be able to recognize Kentucky is ranked first in the nation in overall cancer incidence and mortality rates, with the greatest effect being in the Appalachian region of the state. I would also like the attendees to be able to describe key social determinants of health driving Kentucky's cancer disparities, and to recognize storytelling as an important method for individuals to conceptualize and communicate the burden of cancer around them, as well as to envision culturally tailored interventions. So like I said, Kentucky ranks first in the nation for cancer incidence and mortality, with Appalachia being most affected. And 8% of Appalachians are 8% more likely to die from a preventable or screenable malignancy. Appalachia is also hindered by other things such as high cancer rates, high smoking rates, high obesity rates, chronic infection, poverty, and low education levels. And as you can see, although Kentucky ranks first in the nation for cancer incidence and mortality, it's very localized to this area of the state, which is the Appalachian region. So the culture of Appalachia. I'm from Appalachia, so I can speak to these pretty well. Very rich in storytelling and even tobacco use. It's a very cultural thing And we have strong family and faith connections, which can lead to fatalism because a lot of times we have like an external locus of control. So, you know, I've heard before, if it's my time to go, then it's my time to go. Appalachians are fiercely independent and self-sufficient, which I sometimes call stubborn. This can lead to poor health-seeking practices. And Appalachians value hard work that is often hard manual work, which can lead to exposure to sun and toxins. And vocation is valued over education. So a lot of Appalachians, they want to make money, provide for their families. They value that more than studying out of a book and all of that. And then there's a lot of celebration of tobacco in the coal industry. Growing up in Appalachia, I've seen there's a tobacco festival. There are bumper stickers everywhere that says, "Stop the war on coal, coal keeps the lights on." And I can relate because my father was a coal miner, stepfather, uncles, grandfathers. So coal is it's a valued industry in Appalachia. So for background information, this study is conducted with the ACTION program. And the ACTION program stands for Appalachian Career Training in Oncology. And basically it focuses on research, education, and clinical shadowing to prepare and motivate participants to pursue cancer-related careers. And we also do a lot of outreach. So the ACTION program through Dr. Vanderford and Chris Pritchard actually published a book called The Cancer Crisis in Appalachia, Kentucky Students Take Action. The essays that each student wrote, it detail the effects of cancer in Appalachia. And although cancer statistics are important, data and facts hide the personal stories of how cancer affects individuals and communities. But this book, the whole purpose is to really narrate that, to give cancer more than just numbers, but to show how it truly impacts the community. So my job was to take these 25 essays that students wrote and focus on, well, the essays focused on their experiences with cancer, why they think cancer rates are so high, and what they think can be done to lower the cancer rates. And I used content analysis to identify common themes in the essays regarding types of cancer observed, the causes of the cancer observed, and solutions that could lower the cancer rates. The most common cancer types referenced were breast, lung, and prostate. These were the ones that were most mentioned in the essays. And so the authors found these most impactful, or they noticed them the most in their communities. And then the identified themes that cause high cancer rates were, I could go into a lot of detail on these, but I'll try to keep it shorter. But geography, so, you know, Appalachia has natural resources and this determines the industry. So that's why we have tobacco industry and the coal mines, because we have mountains that are rich in coal and all of that. And then there's also decreased access to healthcare as a result of geography. So for example, like my mama, when she had cancer, she had to drive 2.5 hours one way to get chemotherapy. So there's definitely decreased access to healthcare because of location. And this decreases the opportunity for checkups, screenings, treatment, and all of that. The environment, so a lot of people mention water contamination. A lot of the things mentioned in the essay were, you know, my grandfather said that there's something in the water. That phrase was used very commonly in the essays. So a lot of authors believe that there's water contamination, such as carcinogens from the coal mines, and then chemicals such as arsenic, lead, copper, benzene. And then there's also a lot of reduced accessibility to proper waste disposal and recycling. I've also witnessed this in my hometown. Commonly burned items such as tires, shingles, and building materials, which can contain asbestos and lead. You know, Appalachians love their bonfires. And then there's cultural tobacco use. I sort of touched on this earlier, but it's like a symbol of heritage. It's very culturally accepted, even with its own tobacco festival. And there's just generational tobacco use. So you hear a lot of Appalachians say that they started when they were like 5 because their parents like were fine with it or whatever. So it's very generational, it's passed down. And then poverty leads to decreased health opportunities such as screenings, and have a lot of issues with insurance. And although a lot of people think of like the country or Appalachia being associated with farms and everything, so they would probably think we have plenty of access to healthy food. That's actually not true. There are a lot of food deserts and swamps, so there's elevated consumption of fast food and cheap processed foods, which increases obesity rates. And then education. Low education is an enabler of participation in high-risk behaviors such as tobacco use, prolonged UV radiation, poor diet, substance abuse. And there's decreased awareness of the high cancer rates in Kentucky, cancer symptoms, and recommended screenings. Just a quick example, I came from Hyden, Kentucky, which is about 2.5 hours away from Lexington, and I had no idea that Kentucky was ranking first in the nation for cancer, and I certainly didn't know that the Appalachian region was most affected, but it took me coming to Lexington to find that out as a freshman. And then prevention, there's lack of screenings due to fear, like sometimes they would just rather not know. And then there's appraisal is unnecessary, so it's like, not me, that's not going to happen to me, or my symptoms aren't that bad. And then of course there's mistrust because of cultural differences. So Appalachians, they like to smile when we talk, we like to make eye contact and kind of take our time. Other people, especially in the medical profession, are probably going to be more direct and straight to the point. And sometimes Appalachians can perceive this as rude, so there's just a communication barrier there that causes trust issues. And they also question motives, such as saying, oh, that doctor just wants me to come back for that scan because they want to take my money. I've heard that a lot, and that was referenced many times in the essays. So contributing factors that were most referenced were tobacco use, lack of screening, and cultural influence or cultural adherence. And then the reference solutions were to improve education and awareness, increase screenings, and increase rates of tobacco cessation. But through storytelling, youth learned that data does not tell the whole personal story of cancer's impact. They gained a better understanding of cancer in their communities, and they were able to envision culturally tailored, community-based intervention strategies that could reduce the cancer burden in the area. And here are the acknowledgments. Dr. Vanderford really headed this project, and Lauren and Chris, and here's our funding. And I'm open to any questions. And here are the references.
Very nice presentation, Lauren. While we're waiting for people to type their questions, you mentioned that you have a very personal connection to this type of research, but could you share with us how you actually got involved in the research coming in to UK?
Yeah, so similar to Courtney, I'm from Appalachia, and when I came to UK, someone told me about the action program and Dr. Vanderford, and I ended up joining, and it was one of the best decisions I've ever made. Dr. Vanderford and Chris and everyone in the action program, they've been invaluable for sparking my interest in research. I'm not so interested in like bench research as I am like learning about people, and especially this research is very close to home. It literally is about my home. My mama, just as a personal statement, my mama actually got cancer, and she died before my high school graduation, which she had always dreamed of being at. So when I came to school, I was pretty fresh off of that, and so Dr. Vanderford and Chris had the book published, and afterwards it was so inspiring reading everyone else's stories, and Dr. Vanderford presented the idea to me to perform content analysis on it, and I was super happy to do it. Like, very, very thrilled to be a part of this.
Well, thank you for sharing that. Do you plan to continue doing this type of research, or what are the next steps for you?
I would love to continue doing this type of research. right now I'm actually doing a lot of MCAT studying and all of that, trying to wrap up for graduation and applying to med school in May. So I'm hoping to go to UK if I get accepted, and I would love to continue working with Dr. Vanderford. I'm very passionate about Appalachia and Appalachian research.
Does anyone have any other questions for Lauren? All right, well, fantastic job, Lauren, and hope you get into UK's College of Medicine. Okay. Our next presenter is Irena. And Irena works with Dr. Loria in College of Medicine, Pharmacology, and Nutritional Sciences. Irena, are you ready to present?
Yes.
Okay.
So hi, I'm Irena Antic, and I'm a junior in Dr. Loria's lab in the Department of Pharmacology and Nutritional Sciences. And today I will be talking to you about how perivascular adipose tissue from different depots can influence the endothelial function in obese mice exposed to early life stress in a specific manner. So adverse childhood experiences, or ACEs, are traumatic events that occur during childhood and have been linked to an increased risk of developing chronic disease. Our lab uses a mouse model called maternal separation and early weaning, or MSCW, that mimics adverse childhood experiences and early life stress effects on cardiovascular and metabolic systems, increasing the risk of obesity, hypertension, and cardiovascular disease. We have previously shown that female MSCW mice fed a high-fat diet exhibit a metabolic syndrome-like phenotype and obesity-induced hypertension as they present an exacerbated response to a high-fat diet. Figure 1 shows that male MSCW mice respond to a high-fat diet similar to controls. However, female maternal separated and early wean mice show increased adiposity in response to a high-fat diet. The adiposity is localized on the visceral cavity, and on the left panel, on figure 2, a high fat increases adiposity in female maternal separated and early wean mice. And on the right panel, we see that maternal separated and early wean mice does not increase the amount of subcutaneous white adipose tissue after 16 weeks of a high fat diet, shown here in A and C. However, maternal separated and early weaned mice do increase visceral adipose tissue, shown in B and D. In addition, in figure 3, we can observe that adrenal-derived and circulating aldosterone levels are increased in obese maternal separated and early weaned female mice compared to the controls. We also know that the fat surrounding vessels, or perivascular fat, releases factors that can influence the vascular function and metabolic processes, including adipokines such as aldosterone, corticosterone, and leptin. Therefore, the aim of this study was to investigate whether depot-specific perivascular-derived factors influencing the endothelial function in female maternal separated and early weaned mice fed a high-fat diet act via mineralocorticoid receptor signaling. For the methods, maternal separation was performed by separating the dams from their litters for 4 hours a day, starting on postnatal day 2 to postnatal day 5, and then they were separated for 8 hours a day, starting on postnatal day 6 until postnatal day 16. Maternal separated and early weaned mice were weaned early on postnatal day 17 and fed a high-fat diet of 60% kcal from fat. and control litters remained untouched and began weaning on postnatal day 21 and were also fed a high-fat diet. After 20 weeks of a high-fat diet feeding, both groups were treated with either vehicle or spironolactone for 2 more weeks, and mice were sacrificed at 22 weeks. As for the tissue explants, in order to isolate the tissues, C57B, 6J, female mice were anesthetized with ketamine xylazine and thoracic aortas were dissected. PVAT, or the fat around the aorta, and MSAT, the mesenteric fat around the mesenteric arteries, samples were incubated in DMEM for 2 hours and 37 degrees Celsius. Aortic rings were cleaned and mounted in the wire myograph chamber, and constriction was tested with a dose response curve using potassium chloride. Relaxation was tested on control and maternal separated and early weaned female mice. Both were fed a high-fat diet. Both groups had mice treated with either vehicle or spironolactone, as well as their own perivascular adipose tissue or mesenteric adipose tissue, or left without the explant media and DMEM. Cumulative concentration response curves were performed for acetylcholine after preconstriction with serotonin. And a first cumulative concentration response curve was performed after isolated with rings were incubated with DMEM, followed by a second CCR curve following a PVAT and MSAT media explant incubation. As for the results, we see at the top left graph A that acetylcholine-induced relaxation is similar in both groups when the rings are absent of perivascular adipose tissue. But when we add the perivascular adipose tissue media explant, we see a similar impairment of vascular function for both groups. But in graph B, we see that mesenteric adipose tissue induced an impairment of vascular relaxation that was worse in obese, maternal separated, and early weaned female mice. Next, we see that 2-week spironolactone treatment did not influence vascular relaxation in rings without explant media in either group. And in the bottom left, graph A, we saw that spironolactone treatment improved relaxation in both groups. Similarly, when we incubated the rings in the presence of perivascular adipose tissue media explant. In the bottom left, graph B, we observed that the spironolactone treatment did not induce any further effect on the vascular relaxation when the rings are pre-incubated with mesenteric adipose tissue media explant. So in conclusion, the obese, maternal separated, and early weaned female mice did not show impaired endothelial function of intact aortic rings compared with the controls. And mesenteric adipose tissue from untreated obese female, maternal separated, and early weaned mice impaired vascular relaxation, worsening endothelial function. These data suggest that mesenteric adipose tissue derived factors from female mice exposed to maternal separation and early weaning have a detrimental effect on the vascular function, which could result in increased cardiovascular risk in the long term. Treatment with a mineralocorticoid receptor antagonist improves the endothelial function in both female and control maternal separated and early weaned mice in the presence of perivascular adipose tissue. However, the mineralocorticoid antagonist receptor does not influence the relaxation in the presence of the mesenteric adipose tissue. Thus, these findings suggest that mesenteric adipose tissue derived factors worsening endothelial function in obese, maternal separated, and early weaned female mice were not acting via the mineralocorticoid receptor signaling. And that is it for my presentation. Thank you.
Nice job, Irena. So at this time, we'll take questions if anyone has any. So Irena, how did you get involved in this type of research?
So this was I read about the research actually in an email, and it just sounded really interesting to me. How there was like a mouse model that was mimicking the adverse childhood experiences. And so I contacted the Dr. Loria, and I met with her, and that's kind of how it started. So yeah.
Nice. You do have a question from Dr. Hannan. It's in the Q&A, so let me know if you can see it. If you cannot, I will read it to you.
I have to stop the share to see.
That's fine. You can stop the share.
Or, but do you know if there are any plans to measure their reproductive function in mice? So she's asking if there are any plans to measure the reproductive function of the maternal separated and early weaned mice. Not that I have heard of from Dr. Loria. She has a lot of projects. They looking at fat composition and opioids with the maternal separated and early weaned mice, but none about the reproduction.
Okay, and there's one more from Dr. Bradley.
Are there any other functions to measure? So like a third, something else, like a third factor she likes to look at is low fat diet. That's like another factor, or as function. I know sometimes she focuses specifically, other than vascular reactivity, that's mainly like what she focuses on looking at. And then their metabolic functions and occasionally their brain stems. She also focuses on that.
Okay, well, thank you very much, Irena. Good job.
Okay, thank you.
All right, we will move on to our next presenter. Our next presenter is Cheyenne Chandler. Hi, Cheyenne.
Hi.
Cheyenne works with Dr. Brett Spear in the College of Medicine in the microbiology, immunology, and molecular genetics area. Are you ready to start, Cheyenne?
Yep.
All right.
Okay, let me screen share.
Okay.
To my PowerPoint. Okay. Oh.
I can see that.
Okay, perfect. Okay. I was wondering. Okay, perfect. All right. Hi, everyone. My name is Cheyenne, and I'm under Dr. Brett Spear, and we're in the College of Medicine in the Immunology, Microbiology, and the Molecular Genetics department. And so this research was a further extension of my undergraduate research. And what I did was a characterization of adipose tissue gene expression in the gene AFR2 with mice treated with carbon tetrachloride and black 6 and C3H mice. And so this research was conducted last summer during the pandemic, and it was during the summer fellowship for the undergraduate research office. So for my research, we primarily look into the liver, and the liver in the human body maintains homeostasis, and it also assists in metabolic pathways that ensure that our body influences correctly and there's no issues with anything. And so a failure of the liver can have negative consequences and diseases can arise. Diseases such as non-alcoholic fatty liver disease, NAFLD, in which my lab looks into, reduces liver function, and it is increasing as it is accumulated and associated with obesity. Obesity, as we've heard in the past couple of presentations, is increasing in the United States, especially in Kentucky, and it has increased over the past 50 years. And so for the chart below, you can see that the United States in 1990 was about okay, and then it goes into the 2010s, and you can see that the southern states, especially in Kentucky, has increased over time. So we all live environmental toxins, and one of them my lab uses is carbon tetrachloride, and this can damage the liver. Adipose tissue is outside of the liver, and there's two types of adipose tissue. There's EWAT, which is epidermal white adipose tissue, and there's BAT, which is brown adipose tissue. I primarily looked into the EWATs of this study because EWATs are cells that are lipid storing cells, and this is what accumulates when people become obese. So for the SPEAR lab, they previously identified zinc fingers homeoboxes 2, which is ZHX2, and this is a gene that can influence liver disease. And overall purpose of my study was to broaden ZHX2 targets and identify the clusters that influence NAFLD. And the hypothesis is that there are several gene clusters that do influence NAFLD. So for the methods of my study, I did not actually do wet lab studies. I did the data analysis and I cross-referenced with NCBI databases and other people's gene sets. And so the SPEAR lab previously in like the 2000s range, they did the isolation of RNA, they produced cDNA, and they did the RT-qPCR. And RT-qPCR basically is a technique where you can quantify gene expression patterns and you can look at cell tissue size. So this all changed for my home experiment. And I looked at the different Excel sheets that we wound up getting back. And so there were 3 different genes that they looked at. There was AFR2, ELOVL3, and ZHX2. And so all 3 of these genes are located on chromosome 2. And so we're expecting that a lot of the gene expression patterns will be on chromosome 2. And we looked at 2 different mice strains. One of them is C3H, and the other one is Black 6. And so these 2 mouse cell strains were injected with mineral oil to be the control groups, and the 2 different experimental groups were injected with carbon tetrachloride for the C3H and the Black 6. So looking into the results of my study and what we accumulated through the time last summer, I was looking for the p-value to be low because we would expect that the hypothesis would be true with the low p-value. And we filtered for the chromosome sequence. So we were primarily looking for chromosome 2. And we were looking just to see how many clusters or how many chart records came up and what each gene function did. So for the first group, for the Black 6 mineral oil carbon tetrachloride group, there were a lot of other genes available, but I just did a little snippet because it's just so many genes. And so we found that a lot of the chromosome was 2, and there was a low p-value for this group, and there were 6 clusters. And so this was mitosis, mitotic nuclear division, cell cycle. And so it's basic just regular cell proliferation. And there was next in the far right, there's C3H mineral oil carbon tetrachloride group. And there were 41 clusters that we did identify. A lot of them that we looked into were for chromosome sequence 2, and there was a low p-value. And so this group primarily did cytoskeleton, microtubule, and other cytoskeleton issues and different expression patterns. And so this as well is for cell proliferation. And so this would actually be nice to know and to go further into because we're expecting that there's more cell size and more cell size counts with obesity. And so these 2 could lead into that. And so for the bottom left, it is mineral oil C3 Black 6 strain. And so we saw there was 19 clusters, and we saw there was lipoprotein, myostrate, and cytoskeleton, and these were located on chromosome 2, and there was a low p-value as well. And so for the far right, there was 187 chart records that we did see, and these were for protein binding, transferase, and phosphoprotein, and this is just basic cell functioning. So this was just the bare bones, and there was a low p-value as well. So for the conclusions of my study is that we do believe that AFR2 may have a correlation with ZHX2 expression, and the results that were above do align with the hypothesis that there is a correlation between them both. However, since I did look at old data sets and we want to do more research, we could look into specific gene clusters and see what exactly is going on, and we can find more correlation between ZHX2 and AFR2. So I would really love to thank the SPEAR Lab and the Peterson Lab for helping me throughout all of this, because we did a lot of this research during COVID. And so we all were learning together how to do big data analysis. And so I just really am so thankful for them. So thank you so much and have a great day. If you have any questions, you can let me know.
Great presentation, Cheyenne.
Thank you.
So you mentioned you did this last summer during COVID. Have you graduated?
I am going to graduate in December.
In December. Okay.
Yep.
Awesome. And do you plan to continue? And what are your future plans?
I actually really want to apply to UK for their master's degree program for clinical research. And then I specifically want to work in immunology and microbiology. So I really want to do exactly what I did. So I really enjoyed this because it gave me the opposite side of, instead of wet lab, what are you doing behind the scenes for Excel and everything?
Nice. Did you know when you came in to college that you wanted to do research?
Not really. I knew that I wanted to help people, and I at first thought that I was going to be a doctor. And so I didn't know that I was going to do research, but my major, we're required to do undergraduate research, so I had to. But when I found Dr. Spear, I was like, oh, I love this. And from the moment, I was like, I want to do this for a long time because I just really enjoy it.
That's awesome. Everyone has a different story, and most people don't realize that they want to continue with research until they've actually participated in research, and then like you, they fall in love with it, or they learn that it was a great experience, but they may not want to continue that.
Yeah.
Okay, we do have a question. It is in the Q&A. So let me know if you can see it.
Yeah, I can see it. It's from Dr. Luke Bradley. And he said, great presentation. Which cytoskeleton gene proteins did you look at? I actually can't answer that exact question at this moment. I don't know exactly which actual proteins do that. And so I don't want to tell you wrong. I really don't. But I actually will probably have to get back to you on that. Because I know that all of them work together to really influence each of them. So I would actually have to get back to you for that, Dr. Luke Bradley. Wait.
He said all is good. Does anyone else have any other questions? Oh.
Yeah, there's one from an anonymous attendee. They said, you mentioned that you see a lot of low p-values. What does this mean? And why are low p-values important in this project? So when you have a high p-value, that normally means that you can reject your null hypothesis. And so you basically can say that your study actually did the opposite of what your hypothesis said that it was going to do. And so we were looking for a low p-value because that means that our study was accurate and that all of our data was correlating with each other. And there were some genes that were on the opposite. Some of them were very positive. And so we could actually knock those out because we were primarily looking at what the low ones did and how they influenced the whole entire gene group itself. So low p-values were really, really important in this project because we filtered out immediately all of the high p-values because we were really trying to see the differences between both of them. However, it would be nice to actually go back and then look at the high p-values for the genes that did have a high p-value, because that could also be really telling about how the research was done. So I hope I answered your question. I hope I did.
All right. Fantastic job, Cheyenne.
Thank you. Okay, I'm gonna stop sharing.
All right, thank you. And we will move on to our next presenter. So next is Frances Miller. Franny. Frances works with Dr. Patrick Hannan in the College of Medicine obstetrics. Oh gosh, can't talk. And gynecology. Hi, Francis. Are you ready?
Yes, I'm ready. Go ahead and share my screen.
Awesome.
All right, greetings everyone. Thank you so much for coming here today. My name is Frances Miller, and I'm an undergraduate research assistant in the wonderful lab of Dr. Patrick Hannan, and we are interested in how phthalates impact ovulation. So phthalates are endocrine disrupting chemicals utilized in numerous consumer products such as medical tubing, building materials, cosmetics, and personal care items. And due to their incessant production and utilization, humans are exposed to a mixture of phthalates daily. Phthalates are also known reproductive toxicants that have been shown to disrupt ovarian function. However, the full extent to which they impact ovarian function is currently unknown. So this study will investigate the effects of phthalates on ovulation to better understand their potential repercussions within reproductive health. Previous studies have utilized single phthalate exposures, whereas our study takes a unique approach in utilizing an environmentally relevant phthalate mixture to mimic human exposure. And this specific mixture was derived from a study performed at the University of Illinois, which measured levels of phthalates within the urine samples of pregnant women. And so since this study heavily focuses on ovulation, I hope to provide you guys with some background information into the critical processes that phthalates can potentially affect. So the ovulatory process is occurring as a result of a very delicate framework involving hormones, angiogenic factors, and more. Antral follicles, as you can see here, are essentially the functional unit of the ovary containing the egg or oocyte. And one can visualize how an early antral follicle is transformed into a pre-ovulatory follicle with the help of follicle stimulating hormone or FSH. Ovulation is further stimulated by HCG, which is a potent ovulatory trigger or luteinizing hormone analog that's commonly used clinically to induce ovulation. So after this LH surge, and with the assistance of angiogenic factors like VEGF-A and PGF measured in this study, one can see that the oocyte is released and ovulation occurs around the 12-hour mark in mice. And so once the oocyte is released from the follicular wall, the follicle is further transformed into a corpus luteum. And so angiogenesis, sorry, I'm going to move the Zoom tab. Angiogenesis is also a critical process for ovulation that occurs later in the ovulatory period and is the formation of new blood vessels within the ovulating follicle and transforming corpus luteum. And so LH and HCG bind to their receptor on the granulosa cell, and granulosa cells are the cells that form a layer surrounding the oocyte. So after they bind, multiple angiogenic factors are able to be synthesized, which go on to further mediate angiogenesis. And as a result of their mediation, endothelial cells, which make up blood vessels, are then able to undergo proliferation, migration, and sprouting to form new blood vessels and eventually culminate in a mature vascular network. So we hypothesized that exposure to an environmentally relevant phthalate mixture would decrease angiogenic factors, including vascular endothelial growth factor A, VEGF-A, and placental growth factor, PGF, therefore leading to inhibited angiogenesis. So we mechanically isolated antral follicles from adult CD1 mice and placed them into culture. They were then treated with our vehicle control being dimethyl sulfoxide or DMSO, or the phthalate mixture in a range from 1 to 500 micrograms per milliliter. And so after pre-ovulatory development, the media were then replaced with or without HCG and with or without the phthalate mixture to take further measurements. And so we first measured ovulation rates, where the data here can clearly suggest that phthalates are indeed inhibiting ovulation. And we discovered that increasing doses of our phthalate mixture from 1 to 500 micrograms per milliliter resulted in a decrease in ovulation rates relative to HCG, which you can recognize here in black. And we assessed ovulation by characterizing the oocyte with regards to whether or not it was ovulated out of the follicle.
The treated media were then utilized to quantify concentrations of angiogenic factors, VEGF-A and PGF at 2 ovulatory time points, being 11 hours and 18 hours via ELISAs. And as expected, treatment with HCG did increase VEGF-A concentration levels relative to our DMSO group, which did not receive HCG. However, treatment with phthalates in the 11-hour time point did decrease the concentrations of VEGF-A at 100 and 500 micrograms per milliliter with respect to HCG, and in the 18-hour ovulatory time point at all concentrations from 1 to 500 micrograms per milliliter. Although HCG did not increase concentrations of PGF, the phthalate mixture similarly decreased levels of PGF at 1 through 500 micrograms per milliliter applications of the phthalate mixture in the 11-hour time point, and at 100 and 500 microgram per milliliter in the 18-hour time point. And so ultimately, these data suggest that phthalate exposure is inhibiting ovulation by decreasing the angiogenic factors VEGF-A and PGF, thus potentially contributing to ovulatory defects, which are the leading cause of infertility in women. And so findings from our study can aid in establishing the impact of phthalates on ovulatory angiogenesis and complications in the fertility of women. So I thank you all again for being here with me today. And if you have any questions, feel free to ask them now.
Nice presentation.
Thank you.
So while we're waiting for any questions, Francis, can you share with us how you got involved with Dr. Hannan's lab?
Definitely. So I'm a current junior, and when I was a freshman, I was involved with STEM Cats. And back then we had the in-person undergraduate research showcase. And so when I was presenting my research, I was able to walk around and talk to other groups. And my PI at the time introduced me to Dr. Hannan and his students, and we got to talking just about some of the goals that they had for their lab, and soon came to find out that they were actually looking for another undergrad to fill a position. And I loved the poster, and although at the time I didn't know what everything meant, I just thought it was beautiful and I wanted to learn. And so ever since meeting them at the showcase back then, I've been working since that summer, and it's been amazing ever since.
Fantastic. All right, you do have a question.
Okay, let's see.
And it
The
is in the
VEGF-A levels differ in 11-hour and 18-hour. So Kenan asked, did the VEGF-A levels differ in the 11-hour and 18-hour DMSO group? Oh, so sorry. And so the DMSO, like I said earlier, it represents our vehicle control group, which is pretty much just a baseline in any experiment you want to have a control so that you can compare other treatment groups. And so the VEGF-A levels within these two DMSO groups did not differ, which is represented by these statistical letters. So when letters differ from one another, that signifies that there was a statistically significant difference between the two treatment groups. So between this 11-hour and 18-hour time point, hopefully you guys can see this. Between these two time points, they did not differ, which is represented by this letter A. But great question.
And you have another one.
Yes. What are the phthalate levels found currently? I assume this would be a constant exposure event in daily life, but would a sporadic exposure have the same effects on VEGF and ovulatory processes? So you are correct in saying that it is a constant exposure. And so that is what this phthalate mixture is heavily based upon, being environmentally relevant. We derive that from the urine samples because looking at those urine samples and seeing those levels there, that means that these levels are what is being metabolized by the body. And so when asking, would a sporadic exposure have the same effects on VEGF and ovulatory processes, I think that in that specific moment in time, my hypothesis would be probably not, but down the road, I would definitely expect to see some more dramatic events with angiogenic factors in general, and other potential hormones and other mediators of ovulation. But yes, overall, humans are incessantly exposed to phthalates, so they're a little hard to avoid.
All right, well, thank you, Frances, very much. Great presentation.
Thank you.
Our next presenter is Kenan Flores, who works with Dr. Jessica Blackburn in the College of Medicine, Molecular and Cellular Biochemistry. Are you ready to present?
Yes, I am.
Kenan? Okay.
Hello, my name is Kenan Flores, and over this past semester, I've had the great opportunity of working at the Blackburn lab. At the lab, I was put on an experiment that was occurring prior to my arrival, and this was the testing of different Wnt inhibitors and their efficacy of inhibiting the Wnt signaling pathway. The Wnt signaling pathway is important in cancer research because of its highly activated state in cancers. So a possible method of finding cancer therapeutics is by testing different drugs and seeing if they can inhibit the activity of the Wnt signaling pathway. One way to do large-scale drug screenings is by using mice, but it's difficult because of its high cost to maintain. So at the Blackburn lab, we use an alternative, the zebrafish. And the zebrafish is such a great alternative because they're cost efficient, they're able to produce results rapidly, and they're easy to manipulate in their embryonic stage. One line of zebrafish that is used is called the T-cell factor / lymphoid enhancing binding factor, or TCFLF zebrafish. When the Wnt signaling pathway in these fish are active, genes bind to the TCFLF promoter site, and the promoter site activates gene expression. For our research, our TCFLF fish line drives a reporter gene called green fluorescence protein, or GFP. The reporter protein is important because it's used to quantitatively measure the amount of gene expression. In this case, whenever the Wnt signaling pathway is active, the fish would fluoresce green under a specific wavelength of light, showing the expression. The Blackburn lab completed a screening of over 770 FDA-approved drugs to find compounds that could be repurposed to inhibit Wnt signaling. And through screening, we found 8 novel chemicals that inhibited the Wnt signaling pathway. The protocol in testing the efficacy of each of these 8 drugs consists of 3 parts. The first part of the protocol is testing the inhibition of the Wnt signaling pathway through GFP, or the green fluorescence protein that I previously mentioned. To do this, we first breed the TCFLF line of zebrafish in a ratio 3 females to 2 males. And then we collect their embryos and place them in an incubator for 24 hours. 24 hours after their fertilization, we then screen them. In figure 4a, this is what we'd expect in a normal embryo. There's fluorescence in the tail with the arrow on the left, and there's fluorescence in an area behind their head and the arrow in the right. So we'll choose the embryos that have this type of expression. In figure 4b, this is what the screen would look like under a microscope. So we get rid of the top 2 embryos because they don't have the criteria that we need for the embryos that fluoresce. And for the bottom ones, we would keep those because you can see that their tail fluoresces. And you can see for both the embryos, there's also a dot, which is the fluorescence behind their head. After selecting the embryos that meet the criteria, we then decoordinate them or take them out of their sac using fine point forceps. The decoordinated fish are then placed in a flat bottom 96 well plate, and the desired drugs are added to the plate. After they're treated in drugs for 48 hours, we then use a stereo microscope to take pictures of their tails, and then we use Fiji imaging software to quantify the amount of fluorescence. Results from current testing determined that 4 of the possible Wnt inhibitors significantly reduce GFP fluorescence. These drugs can be seen in the graph in figure 3, where the star range at the top are the 4 drugs that significantly reduced the amount of fluorescence. The second portion of the protocol is testing the regeneration of the zebrafish by amputating the caudal portion of their tail. Regeneration in zebrafish is dependent on the Wnt signaling pathway. The zebrafish used in this portion are 3 to 12 months of age compared to the embryos that were used in the previous portion, and they're also a wild type compared to the TCF-left line that we used in the previous portion. So first, we would anesthetize these fish using a tricane solution for 1 to 2 minutes. This is so that they don't actually feel it when we amputate them, and also so it's easier to amputate them without them moving around. And then after the 1 to 2 minutes are over, they're then placed onto a clean petri dish and they are amputated using a razor blade. In figure 6a, that's what we would expect the amputation to look like. If we amputate more than what is shown, then it could result in increased risk of infection or death of the fish. After amputation, the fish are then placed in tanks that either contain the control or the desired drug that we want. And after 4 days of the drug treatment, we then take pictures of them under a stereo microscope. And then we use Fiji imaging software to determine the amount of tail growth. So in figure 6b, you see the tail is regenerated back normally. And that's what we'd expect for a control fish. But in figure 6c, the tail didn't regenerate fully. And that's what we'd want to see for drugs that inhibit the Wnt signaling pathway. So on the graph in figure 5, we found that 3 drugs inhibited the Wnt signaling pathway and caused a decrease in regeneration. So on the left is the DMSO, which is our control. So we expect there to be regeneration. That's why the fold change is so high. To the right of that is our XAV. This is a solution for our positive control. This is when we expect the Wnt signaling pathway to be inhibited. So we know that it's going to be inhibited, so we know the fold change is going to be tiny. And then that's the same pattern is seen for the 3 drugs to the right of it. And those are the 3 drugs that we found to show similar patterns of inhibition. The third and final portion of the protocol is the eye rescue test. Proper development of zebrafish eyes is dependent on the Wnt signaling pathway. When we overstimulate the Wnt signaling pathway, it results in embryos with an eyeless phenotype. So we can simulate this overstimulation using a solution that we have called bio. So we get embryos without eyes. And we can see if we inhibit the Wnt signaling pathway with these embryos, if we place them in the drugs and they regrow their eyes back. Because using the bio, we overstimulated the Wnt signaling pathway, but when we inhibit the Wnt signaling pathway, they're able to develop their eyes back normally. And we assess these fish by looking at them under the microscope and seeing if they grew their eyes back. So at this time, the screening for each of the drugs is still being conducted. In the future, for the drugs that pass this screening, we'll then test them on human cells that express TCF-left, similar to the zebrafish that we use. And we'll go through the screening process again. And the 2 of the best drugs from that screening will then be used in a leukemia model in mice that we'll make. And the same screening process will be done there. So that is the current state of our project. Thank you all for coming to my presentation. And I'd also like to thank again the people who have helped me throughout the semester at the Blackburn Lab.
Nice presentation, Kenan.
Thank you.
So while we're waiting to see if there are any questions for you very briefly, could you tell us how you got involved in this research?
Oh, yes. So I've always been pretty interested in cancer research and I wanted to get into it. So I'm a freshman currently and I use the resources found at the undergraduate research website, and I looked through other websites and seeing if I was interested in any of their research, and I found Dr. Blackburn and I was really interested in what they're doing, so I emailed her and I got in contact with her over winter break and I got into the lab.
Very nice. Well, nice job presenting.
Thank you.
Does anyone have any yep, there's a question. Let's see, can you see the question, Kenan?
I cannot.
Okay, I will read it to you.
Thank you.
It's from Dr. Luke Bradley. He says, I might have missed this, but what is the throughput of your essay. How many compounds can be tested? How can it scale up and how long?
So currently we're testing these 8 chemicals that we found, and for each part of the protocol, we have different amounts of or fish that we use. And so it'll differ between each part of the protocol, but for how long it lasts, that's also different for each part of the protocol. So let's say for the Wnt inhibitor screen, it would take about 3 weeks to just get through the whole process.
And assay development never ends. Dr. Bradley says, keep going.
Yeah, we are still testing the drugs, and it will take a while, but it'll be worth it in the end.
Well, great job, Kenan. And this concludes our session 3 of first day of our showcase. I just want to say thank you to all the student presenters, and a huge thank you to all your faculty mentors. You all did a great job. So I hope you have a great end to your semester, and congratulations.